research sheds new light on how brain stem cells are activated. Our brains are notoriously bad at regenerating cells that have been lost through injury or disease. While therapies using neural stem cells (NSCs) hold the promise of replacing lost cells, scientists need to better understand how NSCs behave in the brain in order to develop effective treatments. Now research led by the University of Plymouth helps to shed new light on the mechanisms used by NSCs to wake up, going from their usual dormant state to one of action. NSCs produce neurons, nerve cells, and surrounding glial cells in the brain. By understanding how NSCs work, it could pave the way for therapies to speed up the neurons and glial cells regeneration. The new study, conducted using Drosophila fruit flies, shows that molecules that form a complex called STRIPAK are essential to promote reactivation in NSCs. STRIPAK, striated interacting phosphatase and kinase, is found in organisms from fungi to humans, and the team uncovered it when comparing the genetic messages of dormant and reactivated NSCs in live fly brains. The researchers then discovered that STRIPAK components act as a switch to turn off dormancy or quiescence and turn on reactivation. Lead author Dr. Claudia Bajos, from the Institute of Translational and Stratified Medicine at the University of Plymouth, acknowledges there is still a long way to go until such findings can be translated into human treatments. But she explains the significance of the new work. So little is currently known about how neural stem cells coordinate cues to become active and direct the production of more brain cells, she said. These stem cells last throughout life mainly in a dormant state, so learning how they work is critical to our understanding of cell regeneration. This study reveals that STRIPAK molecules are essential to enable reactivation in NSCs, and we are very pleased with the outcomes. But we are only at the beginning. We are working to expand our findings and bring us closer to the day when human neural stem cells can be controlled and efficiently used to facilitate brain damage repair, or even prevent brain cancer growth that is fueled by stem-like cells. The full study, entitled STRIPAK Members Orchestrate Hippo and Insulin Receptor Signaling to Promote Neural Stem Cell Reactivation, is available to view now in the journal Cell Reports. The work was supported by the University of Plymouth, Faculty of Medicine and Dentistry, the Leverhulme Trust, the Biotechnology and Biological Sciences Research Council (BBSRC), the DFG German Research Foundation and the Johannes Gutenberg University, Germany. The work of Dr. Bajos and her team takes place within the Brain Tumor Research Center of Excellence at the University of Plymouth. Choosing the right drug to fight cancer. Canadian researchers have discovered a molecular indicator of a mechanism that drives cancer progression, giving doctors the possibility of using precision medicine, that is, choosing which patients will respond to a particular anti-cancer drug. In a study published in Cancer Research, a team of biochemists at Université de Montréal found that a group of enzymes called SRC kinases chemically modify a tumor-suppressing protein called SOCS1. SOCS1 is part of a gene regulation circuit centered around the master cell proliferation regulator p53, often called the guardian of the genome, said senior author Gerardo Ferber, an UTM biochemistry professor and researcher at its Hospital Research Center, the CRCHUM. If p53 or another protein in its network is mutated or becomes chemically modified in some abnormal way, a pattern of gene activation occurs that programs cells to proliferate without control, as occurs in cancers. In their research, led by UTM PhD student Emmanuel Saint-Germain, with UTM biochemist Frédéric Lessard and Université de Sherbrooke biochemist Subbaraj Ilangumaran, Ferber's team uncovered a new mechanism by which the p53 circuit becomes unbalanced. Normally, the SRC kinases add phosphates to proteins in a cell in a highly regulated manner. But in cancer cells the regulation of these enzymes can break down. As a consequence, SOCS1 is abnormally targeted by these enzymes, leading to an effective inhibition of its ability to regulate p53 and stop the proliferation of cancer cells. The therapeutic implications of UTM scientists' discovery could be multiple, they believe. Since effective anti-cancer drugs that target SRC kinases already exist, detection of modified SOCS1 in a tumor could be used to predict whether these drugs would be an effective treatment for the tumor. We were able to detect phosphorylated SOCS1 in patients' samples with an antibody that we developed, said Saint-Germain. The same antibody could be used to detect phosphorylated SOCS1 in a clinical setting, providing a way to decide whether SRC kinase inhibitors would be an effective treatment. Added Elangumaran, who has been studying SOCS1 in immune cells and cancers for many years, this new mechanism for SOCS1 inactivation may actually represent a regulatory control that is hijacked by cancer cells. On a more fundamental level, our group's discovery that phosphorylation of SOCS1 acquires a new physical form opens the door to hitherto unknown ways of regulating SOCS1 functions. And this has implications for the treatment of autoimmune diseases and for anti-cancer immunity. Newfound autoimmune syndrome causes muscle pain, weakness. A previously unknown autoimmune muscle disease involving sudden onset of debilitating muscle pain and weakness has been identified by researchers at Washington University School of Medicine in St. Louis. The syndrome easily could be mistaken for other muscle diseases that require different treatment, so the findings are expected to help physicians treat patients appropriately, the researchers said. We observed only 4 patients in more than 20 years, so it's very rare, said senior author Alan Pastronk, MD, a professor of neurology, and of pathology and immunology. It has never been described before, and it seems to be treatable, so it will be helpful for physicians to be aware of it. People don't need to be afraid this is going to happen to them, but in the extremely rare event that it does, physicians should now have the tools to respond. The findings are available in the April 6 issue of the journal Neurology. In 1996, Pastronk, who leads the university's neuromuscular disease clinic, was reviewing microscope slides of muscle from a patient with muscle pain and weakness when he saw something odd. Scavenger cells that normally feed on dead material were crowded around injured muscle fibers. This was an unusual enough situation that I kept it in my memory and stayed on the lookout for other cases like this, Pastronk said. Over the next 22 years, he encountered 3 more patients hospitalized for muscle pain and weakness whose biopsies showed muscle breakdown coupled with scavenger cells. By carefully analyzing the biopsies, Pastronk and colleagues discovered that the patient's own muscles were under assault by immune scavenger cells known as histiocytes. They named the newfound syndrome large histiocyte-related immune myopathy. The disease also reflects a new way in which the immune system can damage muscle cells. Understanding how muscle fibers are damaged by immune cells could lead to ways to prevent the damage. There are several immune muscle diseases where the primary damage you see under the microscope is muscle fiber breakdown. Each of those diseases has a different prognosis and requires a different treatment, Pastronk said. Physicians need to recognize the immune nature of this condition in order to treat it appropriately. 3 of the patients improved when treated with immunomodulating therapy such as steroids and have not relapsed since. The 4th was undergoing cancer treatment when the muscle symptoms developed, and he died due to cancer shortly afterward. Further, while muscle pain and weakness brought the patients into the clinic, all 4 were also anemic and had other shared symptoms, so accurately diagnosing people with the syndrome would help doctors manage and treat their other symptoms as well, Pastronk said. The condition also could be mistaken for rhabdomyolysis, a disease in which muscle fibers break down due to injury, overexercise or other reasons. This syndrome generates results similar to rhabdomyolysis on a blood chemistry workup. But people with rhabdomyolysis usually recover on their own, whereas the new syndrome can be effectively treated with anti-inflammatory drugs. Alternative molecular mechanisms observed in cancer cells. Current anti-cancer drugs can be quite effective, but too often, tumors are not fought off completely and end up returning. A recent study published in the FASEB journal provides the first evidence that some cancer cells evade therapy by switching over to alternative molecular mechanisms that are not affected by existing anti-cancer treatments. A research team from Innate Repair, a biotech venture of Technische Universität Dresden that develops novel therapeutics to fight cancer stem cells, conducted the study. To test their hypothesis of alternative molecular mechanisms or cancer cell escape routes, researchers used cultured cells from brain cancer patients. They placed the cells in culture media designed to maintain them in 3 states, one where the cells utilized the established mechanisms to grow, one where they used the alternative mechanism, and one where they utilized both. The researchers found that when the alternative mechanism was used, the cancer cells behaved very differently from when it was off. The cell's mechanical properties, shape, size, deformability, gene expression properties, i.e., which genes are turned on and which are turned off, and susceptibility to treatments changed dramatically. By modeling this alternative state, researchers were able to identify several treatments, both new to human use, as well as drugs already approved for other conditions, that specifically kill cancer cells in this alternative state. To kill the same cancer cell in one state, you need very different drugs than to kill it when it is in the alternative state, said Andreas Andrautselis Theotokos, Ph.D., CEO of Innate Repair. By focusing on those drugs that only kill cancer cells when they are using the alternative mechanism, we identified several drugs that, so far, probably have gone unnoticed. Andrautselis Theotokos and colleagues are hopeful that the drugs they identified that are already approved for other conditions could be repurposed in oncology relatively quickly, providing new options to cancer patients. In addition, their findings could potentially extend beyond brain cancer to cancer in general, as their prior work has demonstrated that this alternative molecular mechanism is also operational in several other types of cancer. This is a provocative lead that, if translated to animal studies, could lead to new drug development paradigms in oncology, said Thoru Peterson, Ph.D., editor-in-chief of the FASEB journal.